The first structure of proteins we should mention is the primary. Sometimes we'll call this like so. This is the linear sequence of the protein. So you'll have your NH3, then you'll have a string of amino acids, met, his, glutamic acid, arginine, and then your carboxyl group. This is the primary sequence. It is defined, or held together, I should say, by these bonds, and these are called peptide bonds. Peptide bonds. The primary structure is non-functional. Next, let's talk about our secondary structure. And we'll often just call it like that. It's important to remember before we talk about the secondary then the tertiary is that even though this primary doesn't have a function, its sequence helps dictate how the protein will fold into secondary and tertiary forms. The secondary we'll call regional folding. And it will form one of several kinds of secondary structures, but the two you should know include the alpha helix and the beta sheets. You won't have to draw these, but if you look in your textbook you'll see examples of them and you should be able to recognize them. The alpha helix is just something like this, and the beta sheet we often draw like this. So the protein, any given protein may have several different secondary structures within it. It is often characterized by, or held together I should say, by hydrogen bonds. So hydrogen bonds are really important. Now, I'm going to write usually non-functional. Say usually because certain proteins in a secondary structure have a function, and the most important one that you should probably know are the prions. If you've heard of prion diseases, these are like the mad cow disease or chronic wasting disease in deer that sometimes we'll see in Michigan. These proteins don't fold beyond the secondary. They only fold by into the secondary structure. And they're infectious. One of the few agents that are infectious that are just a protein, not a virus or anything like that, just a protein. Okay, let's talk about the tertiary structure next. And I'm going to erase some of this here just to give me some room. The tertiary structure, sometimes written like that, this is the fully folded protein. It is certainly functional. And you've seen me before when I write a protein structure, I'll usually draw it as this kind of mass here. And that's usually what happens. They fold upon themselves. The secondary structure still pretty much, when you look at it, looks like a linear array of sequences, though there is some local folding. This looks like a glob of amino acids, but a very functional glob of amino acids. It is primarily characterized by hydrophobic forces. And we want to come back to this when I have a little bit more room to write on the board, but we'll definitely come back to it because it's one of the most important features of a protein's function. Okay, and then the last one we want to talk about are the quaternary structures. And while I'm at it, I should point out that every single protein has a primary structure and a secondary structure. Almost all proteins have a tertiary structure. The notable exceptions would be the prions. However, not all proteins have a quaternary structure. And I'm running out of room here, but let's draw the hemoglobin molecule we drew before, where we had the two alpha subunits here and then the two beta subunits here. So a quaternary protein is composed of multiple subunits, often coded for by different genes. They are then held together to give that protein function. For instance, the hemoglobin, any one of these subunits by themselves, is not functional. These are often characterized by ionic bonds, hydrogen bonds, I'll write it here, we call these disulfide bonds. There can be some hydrophobic interactions as well. So a lot of things can hold these quaternary structures together. Not covalent bonds, but they're more weaker associations. Van der Waals would fit in here as well. Now I want to come back to hydrophobic forces, but I need to erase all this first. It's important to note that every single amino acid has a similar structure. We have an amino group here, we have a carbon here, and then we have a carboxyl group at the end. We also have a hydrogen here. Now what makes an amino acid different from another amino acid is what we call this R group. This is the functional group. Now this R group makes that amino acid fall into one of a few categories. That R group could make an amino acid nonpolar. It could make it polar. It could make it aromatic, meaning it has a ring structure to it. These polar amino acids could either be neutral, like I wrote here, or I'm writing here. They could also be polar acidic, which means they have a negative charge on them. They could also be polar and they're basic, meaning they have a positive charge on them. And your book mentions some of these non-standard amino acids, but we're not going to worry about those. It's important for you to know that these 5 groups of amino acids exist. You don't need to know which amino acids fall into these categories. I suspect you'll learn that in biochemistry if you take biochemistry. But for now, you should know that these 5 exist. You should also know where they would be at in a protein, particularly a folded up protein. So we'll draw one in its tertiary structure. In this tertiary structure, remember I mentioned that term hydrophobic forces. Nonpolar we can also think of as being hydrophobic. We can think of the polar ones over here as being hydrophilic. So our hydrophobic ones are going to be in the middle of that protein. They're trying to, for lack of a better way of saying it, move away from the water. They're avoiding that water, so they're going to be in the middle. So I'm going to put a 1 here, in the middle of the protein, not exposed to the outside of the protein. I put a 1 there because that matches up over here. On the outside of the protein, that's where we're going to find, on the outside of the protein, that's where we're going to find our polar amino acids, particularly our acidic and basic ones. So I'm going to put 4s around here. And that's because they need to interact with water and other molecules. For instance, when it forms a quaternary structure, these polar amino acids may be what is linking the 2 subunits together. So it's very important to know nonpolar amino acids are in the middle of the protein. Polar amino acids are on the outside of the protein. While we have this beautiful drawing written here, let's take this opportunity to explain why we begin in a protein sequence with NH3 and we end it with COO-. As this amino acid chain is growing, they're added to this COO-. So the next amino acid that is attached here binds here. And you keep doing this till you get to the end, and when you add that last amino acid to this protein sequence, it's always going to have that COO- at the end, the carboxyl group at the end. We don't add to this end, so this is always going to be at the beginning of a protein. Okay, this was a particularly longer podcast, but I think all of this kind of tied nicely together, so I didn't want to break it up, but I think we are done with it. So let's close with a quick summary. You should know the various features of the genetic code. We talked about several things. We talked about what the nonsense codons were, what the sense codons were. We talked about the 3 nonsense codons, also called stop codons. We talked about the start codon. We talked about some features of the genetic code. We talked about it being universal. We talked about it being degenerate. We talked about it coding for synonymous amino acids. We talked about a few exceptions, and most of those exceptions were found in specific mitochondria, as well as protozoa and some archaea and bacteria. But you should also appreciate that the code is, by and large, very universal. We also talked about how that wobble effect allows the code to be degenerate. Next, we talked about reading the messenger RNA to make a protein. And remember that we had this long nucleotide sequence, and we had to get past that 5' untranslated region. You should also know why that's important. And then we looked for that very first AUG. And once we found that AUG, we started plugging in amino acids until we got to one of the 3 stop codons. Once we got there, we ended that sequence with a COO-, a carboxyl group. And again, remember the front of it is an amino group. Like so, with all of the amino acids here in between. Then we ended with talking about protein structure. Talk about the 4 different forms: primary, secondary, tertiary, and quaternary. You should know which ones exist in all proteins, or almost all proteins. You should know which kinds of chemical bonds characterize that particular level of protein folding. And then you should know the basic structure of an amino acid that we talked about very end. So we'll say the last thing here, amino acid classification. That is that amino acids are either polar, nonpolar, aromatic, or polar neutral. And that these characteristics allow that protein to fold in a proper way, where we have charges on the outside of the protein and hydrophobic or nonpolar amino acids on the inside of that protein. Well, that ends this podcast. If you have any questions at all, please make sure you come talk to me. If not, I'll see you in class. Bye.